Hello, everyone. Myself and Diagnostic Solutions Laboratory want to thank you so much for taking the time to be here with us today. My name is Dr. Natalie Groenewoud, and I am a naturopathic physician. I've been in practice for over a decade and have a strong passion and focus for women's health and digestive health. And I'm also an educational consultant here with Diagnostic Solutions Laboratory. So I'm feeling really honored and privileged to be able to provide you all with a three-part lecture series all about the normal bacterial flora. And as a clinician, I always appreciate lectures that really provide me with some clear, digestible and applicable information. So my biggest goal throughout this lecture series is to provide exactly that for you. So what you can expect for the next three lectures, the first one being today we're going to discuss all the roles of the normal bacterial flora, how they help our GI tract health and our overall health. Lecture 2, I'm going to discuss the section on our GI map that is all things normal bacterial flora and what it means if those species are high or low, what's potentially causing those elevations or deficiencies, and then potential treatment options. And then the third lecture is going to be all about case studies. So this will be a really nice way for you to observe how I interpret the normal bacteria flora section on the GI map in conjunction with the overall GI map and the patient's presentation, and kind of bringing that all together to provide some treatment options or considerations. So let's get started. I think, normal bacterial flora, we are learning so much about this frontier daily. There's new research, there's new insights. It's a very exciting time to uncover all things normal bacterial flora in nature. And I think it's really exciting for us in particular because we've got such an intimate relationship with normal bacterial flora. So as you probably all know, over millions of years of coevolution, we've been able to develop this really beautiful synergistic, mutual and beneficial relationship between ourselves and our microbiota. And so our microbiota helps a ton with our different physiological functions, and we in turn are able to offer them nourishment and a place to live. So here's a list of everything that we're going to talk about today in terms of how the microbiota are going to help with these certain areas of our GI tract health and our overall health. So let's dive into the epithelial barrier function. I don't know about you, but it always blows my mind that between our immune cells and the lumen of our intestinal tract, which is really exposing us to our external world, in the form of food consumption, that protective layer is only a single cell layer thick. So it makes complete and utter sense that is only a single cell layer when it comes to needing to absorb all the nutrients from the food that we are eating. But it starts to feel a little bit vulnerable when you're considering, potential pathogen exposure or potential toxin exposure. And this is where our nice, commensal beneficial bacterial flora are really going to support us in beefing up that security at that epithelial intestinal layer. So not only are they providing us a physical barrier, the commensals, as you see here, make up part of that mucosal firewall. So they are physically going to make sure that, no pathogenic organisms are able to get through that mucus layer and onto our epithelial cell layer. But not only do they provide that physical barrier, they're also producing short-chain fatty acids. And those short-chain fatty acids help in a couple different ways too. So short-chain fatty acids can actually increase transcription of mucin from the goblet cells. So that's going to make this mucus layer nice and healthy, nice and robust, again, preventing any sort of microorganisms from getting into contact with the epithelial layer. And then different short chain fatty acids also are able to facilitate tight junction assembly. Again, really important to just make sure that intestinal cell layer is nice and tight and not permeable because we don't want any intestinal permeability to make our immune system more vulnerable to infections. So a couple things to consider when it comes to treatment. Just remembering that if you do see on your patient's profile low commensals or deficient beneficial bacteria, that there probably is some epithelial barrier function too. So just having that in mind that's going to need some extra care and consideration. And then also, if the patient's zonulin is elevated, and that's also tested on our GI map as an add-on. So if their zonulin is elevated, that you may want to consider probiotics in that situation too, just to help to support that tight junction assembly. All right, let's move on to how our commensal bacteria flora help with the development of our immune system. So under normal conditions, the fetal gastrointestinal tract is actually sterile. So the first exposures that we start to get to the microbiota is coming through the vaginal canal. So our mothers, or that maternal vaginal flora, helps to wash over our bodies, and then that starts to seed some of these beneficial bacteria. These exposures will continue with the consumption of colostrum and breast milk. And so these early interactions are really important to just set the tone of long-term mucosal and systemic health. So obviously this is not everybody's process. For whatever reason, vaginal deliveries and breastfeeding are not always possible. So if this is a situation for you or your patient, a treatment consideration just to get that early exposure would be to give the maternal lactating mother or the newborn some Lactobacillus acidophilus and Bifidobacterium infantis, and that will help with that early exposure to some microbiota. So the importance of the early microbial exposure is really driven home and can be clearly identified when we start to study animals that have been raised in a germ-free environment, and then assessing how this has affected the development of their immune system. So animals that have been raised in a germ-free environment, they have noted that they start to have, or that they have poorly developed GALT, or gut-associated lymphoid tissues. And so we are starting to see, they'll have fewer T cells, dendritic cells, weaker antibody responses, and impaired oral tolerance. So that's pretty significant. So not only do these commensal bacteria really help with the development of our immune system, but after we have that initial exposure and the development has taken place, the commensals are also going to help with the modulation of our immune system. So the GI tract is the primary site of interactions between microbes and the host immune system. We are essentially combining 100 trillion microorganisms in the GI tract, which also harbor 70% of our body's lymphocyte population. So it's a lot to cram into a little space, and this is making it a really significant immunological organ in the body, and making it the largest one, actually. So let's take a look. In terms of our gut-associated lymphoid tissue, there's going to be 4 main specialized immune cells or tissues that are found in the gut. And I'm just going to chat through how your normal bacterial flora can support different immune cells in these areas of the gut, and how that's going to help with modulating our immune system. So we'll start with the Lamina propria lymphocyte. So these lymphocytes are scattered in the lamina propria of the mucosa, and the majority of them are IgA secreting B cells. So there seems to be an interesting and synergistic relationship between our commensal bacteria and secretory IgA, and it looks a little bit like this. So the commensal bacteria are going to stimulate the maturation of our GALT, which in turn will help with the production of our IgA from our B lymphocytes. And then the IgA, the secretory IgA, coats the microbiota, which allows the commensal bacteria to move into the GALT without being seen as an invader. And then from there, our host immune system can govern the quantitative and qualitative composition of our commensal microbiota. So really, our commensals are helping to produce that secretory IgA. The secretory IgA is then mounting on to the commensals, allowing it to come and be identified by the immune system, and then the immune system can kind of decide what kind of milieu it wants, what type of commensal bacteria it wants, and how many. So interesting relationship there for sure. And some therapeutic considerations are if secretory IgA is low, again, another marker we find on our GI map to consider supporting that patient with some probiotics or commensal bacteria. All right, looking at a couple other immune cells, we'll move towards the macrophage. So macrophages are specialized cells involved in the detection, phagocytosis, and destruction of bacteria and other harmful organisms. And short-chain fatty acids from our beneficial bacterial flora really drive the differentiation of a monocyte into a colon macrophage. So those short-chain fatty acids are really important to help with that differentiation process. The other thing that short-chain fatty acids do is they are one of many signals known to be able to drive an antimicrobial phenotype in a macrophage. So they really help to produce, characterizing that macrophage as more of an antimicrobial type. So it is interesting though, these commensals have this nice relationship with our immune cells, but with providing food for these immune cells, but if they were to translocate across the epithelial cell barrier, these macrophages would rapidly engulf them and eliminate them. So there is this continuous dance that exists between our microbiota, the intestinal epithelium, and our immune system. On one hand, the intestinal epithelium and the gut are able to benefit from a large array of these short-chain fatty acids that the commensal bacteria are producing. And on the other hand, the microbiota still kind of need to keep them at an arm's length from the epithelial barrier to prevent any setting off multiple immune complexes that can trigger other immune reactions, including some sort of autoimmunity. So it's almost like, okay, thank you so much for your food, but keep your distance at the same time is a relationship between the macrophages and the commensal or beneficial bacteria. All right, so then we're moving on to the Peyers patches. And Peyers patches are essentially immune sensors of the intestinal tract. They contain these dendritic cells. They're antigen presenting cells. And essentially what they do is they have these little pseudopods that are going to go into the lumen and really get a sample of what the environment in the lumen is like. I kind of think of them as wine tasters. They go in, try a wine, decide whether they like it or not. Remember the name of the good ones, and then if they don't like it, getting rid of the bottle of bad wine that they had. So the dendritic cells are essentially doing the same, sampling the microbes and deciding which ones they like or don't like and want to discard. Our beneficial bacteria activate dendritic cells, so that helps to induce the development of Treg cells, and that in turn can control our Th1, Th17. So really important in getting adaptive immunity and protection against infections here. So a couple of therapeutic considerations. If your patient's commensal bacteria are low, don't be surprised if they're experiencing some immune dysregulation. And then also if your patient is having some immune concerns, an assessment of your commensal bacteria could be really insightful to see what's going on there. All right, moving on. Our commensal bacteria also produce anti-microorganism substances. So not only can they produce them themselves, but they can also stimulate the gastrointestinal epithelium to produce these antimicrobial peptides. And so there's a couple examples here. So the commensal bacteria, again, can support or trigger the epithelial cells to produce B defenses. So lactobacillus in particular can stimulate the production of B defenses in the human epithelial cells. This can also be done directly by the commensals themselves. And I think that this is really cool. So E. coli produces bactericidins. So that commensal species can produce bactericidins, impairing the growth of related pathogen enterohemorrhagic E. coli. And the same is true for staph, so Staphylococcus epidermis and Staphylococcus aureus. So really neat to think about in terms of therapeutic considerations. If you're finding a pathogenic strain on your patient's test, and this is, this can be found with our GI map. So we've got the pathogenic E. coli on page 1, and then we've got the Escherichia on page 2 under the normal bacterial flora. So, if the pathogenic strain is present, how can we build up that commensal species that's in the same family to help produce those bactericidins against the pathogenic strain? So really cool and really interesting considerations for your treatment plan. So the commensal bacteria are also very, very great at inhibiting pathogen adhesion. So probiotic strains have great adhesion ability and can block the adherence of pathogens just competing for those host cell binding sites. So I think this is really good optic here to see, if we don't have enough probiotics and those pathogens can start to adhere to these attachment sites. If we have a nice volume of probiotics, that makes it a lot harder for these pathogens to start to adhere to those reception sites. So this is going to help with preventing any sort of infection, just that competitive inhibition there. So that's really, really quite a simple thing to think about. The other thing is that our microbiota can also competitively exclude pathogenic microorganisms. So the principle of competitive exclusion just means that two species cannot coexist if they occupy the same environment. And several examples of this can be found in the literature. So clinical trials have shown effectiveness of Lactobacillus rhamnosus against vancomycin-resistant Enterococcus. Another one, an in vitro study, showed L. reuteri co-aggregated with Candida albicans during biofilm formation, creating a hostile environment that inhibited the yeast overgrowth. Colonization of strain Bifidobacterium longum that produces high levels of acetate conferred protection against lethal enteropathogenic E. coli. So just thinking about this, there's certain strains that just can't coexist in the same environment. And if we can bump up the beneficial bacteria, that is going to really outcompete a pathogenic bacteria, how fabulous. So again, here are just a few examples. I am sure there is tons more in the literature for us to find, but just even thinking, if enterococci is on your patient's panel, think about adding some of that Lactobacillus rhamnosus to outcompete it. So we can start to get really creative with our treatment plans. All right, so that's talking a lot about how our commensal bacteria really help our GI tract health and immune health there. But they also are going to help with producing energy products or energy production substances for our epithelial cells too. So they're really making sure that the epithelial cells are really taken care of as well. So short chain fatty acids are a major energy source. Butyrate specifically is the main energy source for colonocytes. And primary colonocytes consume more than 70% of oxygen due to butyrate oxidation. So that's just showing that energy pathway for these colonocytes. And an energy deprived state leads to lower ATP levels and ultimately autophagy of the colonocyte. So if we don't have that nice butyrate in our intestinal tract, that can be very detrimental to the colonocytes and actually cause them to not survive. And then we're going to have to deal with some intestinal permeability. So A couple therapeutic considerations. One is just eat your fiber, feed those beneficial bacterial flora so that they can produce those short chain fatty acids to feed your colonocytes. The other thing you may want to consider is supplementing with butyrate, especially if your major butyrate producing species are deficient. So just some insight, Clostridia, Faecalibacterium prosnitzi, and Firmicutes, those are all major butyrate producers. So if you're seeing those low on the GI map, give your patient some butyrate until we can get those strains in a healthier population size, just so that we are feeding those colonocytes the nutrients they need to have the energy they need to survive. All right. So our commensals are also responsible for producing vitamins. So vitamin synthesis, a great example of how our commensals are responsible for vitamin synthesis is seen in ruminants who have no dietary requirement for water-soluble vitamins. And then on the contrary, animals, lab animals that are in a germ-free environment do require those dietary supplementation of vitamins. And that's just based on the microbial populations that these animals have in their intestinal tract is providing them with that vitamin synthesis, whereas the germ-free animals don't. So several bacterial genera in the distal intestine, so your Bacteroidetes, your Bifidobacterium, and your Enterococcus are known to synthesize vitamins. So your thiamine, folate, biotin, riboflavin, and pantothenic acid. Those are all going to be produced in your intestinal tract by your commensals. It is also been established that up to half your daily vitamin K requirement is also made by your gut bacteria. So a couple of therapeutic considerations is if you or your patient have low beneficial bacteria, it may warrant giving them, some vitamin B supplementation and some vitamin K, especially if they're experiencing low energy as well. All right. Commensal bacterial flora are also really important in supporting our metabolism. So what we've seen is a genetic sequencing study identified that obese animals have markedly decreased abundance of Bacteroidetes and an increase or an abundance of Firmicutes and methanogenic organisms. So what is it about the Firmicutes that is really promoting this obesity? We've been able to see that the microbiota of obese individuals, so that Firmicutes, has an increased capacity to harvest energy from the diet. And they did some I'm going to say this wrong, calorimetry analysis, and that showed that there was increased concentrations of short chain fatty acids and significantly less energy remaining in the feces of obese mice relative to their lean counterparts. So essentially, the Firmicutes are extracting a higher caloric density of the foods, and that's making them more prone to obesity. So some therapeutic considerations, if you are seeing an elevated Firmicutes, a lower Bacteroidetes, or an elevated ratio, is that alterations in the microbiome is reversible. So the ratio of Firmicutes to Bacteroidetes species will decrease over time if your patients go on a fat-restricted or a carbohydrate-restricted diet. All right, so how do we analyze kind of an overall state? The next lecture I'm going to go over more specific species, but how do we analyze our overall state of our commensal bacteria flora? And essentially on page 2 at the bottom, you will see the phyla-microbiota listed. And 90% of our gut microbiota falls into these two phyla, the Firmicutes and the Bacteroidetes. So this will give us a really nice overview of how our normal bacterial flora are doing as a population. And when you're viewing your patient's tests, and you do observe this overt deficiency at the phyla level, you now have this understanding of how gravely important it is to get this population of microbes back up to a healthier size, because they're just so, so important for our GI health, our overall health, um, as we've identified today. So I'd just like to conclude today's presentation by saying, the human body, we have been so innately wise to evolve with these microbiota with time, and this intimate relationship that we have with them is really important to nourish. So the definition of commensal means to actually sit together at the same table. So I think it's really important for us to educate ourselves and our patients that, our job is to be excellent hosts. We need to be eating a really healthy diet, lots of fiber in it to feed them. We want to limit the use of antibiotics and exposure to toxins that really depletes them. And we want to give them a nice, happy, healthy home to thrive in. Thank you so, so much for spending this time with me today. I am so looking forward to continuing this conversation with all of you. And I just want to outline what's coming up next. So if you're still with me here, thank you so much. The next lecture, again, we're going to be going over all of the normal bacterial flora on page 2, going through each species, showing you what it means when these are high or low, and some treatment considerations. And I think that this is really important. A lot of practitioners get a little bit stumbled here because we're so used to wanting to see below detectable limit on all the other areas of the test, because we're looking at pathogens and we're looking at dysbiotic bacteria. But the normal bacterial flora, highs and lows really mean different things. And of course, we don't want to see any of our normal bacterial flora below detectable limit. So please stay tuned for this next lecture, and I hope you have an absolutely wonderful rest of your day.